Yeah, welcome to another session of Bioinformatics. I am Dr. Y.V. Rajesh, Department of Biotechnology, KL University. In today's session, we will learn about methods for multiple sequence alignment. So, in previous sessions, we learned about basics of multiple sequence alignment. So, what is the need for multiple sequence alignment and how multiple sequence alignment can be done, such as local alignment and global alignment. In today's session, we will learn about algorithmic approaches. So, what are the different methods through which different programs that we can use for achieving multiple sequence alignments. So, basically there are three important algorithmic approaches are there. So, dynamic programming approach, which are also called as exact methods, and the second one is progressive alignment method, and third one is iterative approaches. So, there are other methods also there, structure based alignments, methods which are for only specific cases, and we have hidden Markov model based alignments. So, 5 different type of approaches are there: dynamic programming, progressive alignment, iterative approach, structure based, and hidden Markov model alignments here. So, at our level, at undergraduate level, so we'll focus on dynamic programming and progressive alignment only, but just we'll learn that other methods also available here. So, first we start with dynamic programming. So, in pairwise sequence alignment, dynamic programming is something like this. So, we take two sequences, one on horizontal axis and another sequence on vertical axis, then we using gap scores, match score and mismatch scores, we construct the matrix, then we fill the matrix, and whatever the score we get, that we'll call it as a final alignment score. So, we have Needleman-Wunsch algorithm, Smith-Waterman algorithm for dynamic approach of pairwise sequence alignment. The same approach is extended or extrapolated to multiple sequences. So, here instead of 2 sequences, we will take 3 sequences. Assume that we have 3 sequences. 3 sequences will form sides of a cube, and if you have multiple sequences, then hyperdimensional elements or hyperdimensional spaces are constructed and alignment is done here. So, when you have 3 sequences A, B and C, suppose A indicates some ATGCT, B indicate ATGCA and C indicate ATGGG. Assume the 3 sequences are there. Then the dynamic approach, simplest dynamic approach, what it will do is, so it will take one sequence on one side, another sequence on another side, and another sequence on another sides of a cube or cuboid. Then pairwise alignment is done. So, first B and A pairwise alignment, then B and C pairwise alignment, A and C pairwise alignment, it is done. Then, so based upon this pairwise alignments, an optimal alignment is achieved here. So, we will see an example for this one. So this is an illustration indicating that 3 pairs of alignments. You have green color, cyan color and orange color. So, 3 pairs of alignments. The final result will be grayish color path or trace that we can see in the final alignment. We see an example. Assume that I have 3 sequences, D, Q, L, F, sequence 1, and another sequence D, N, V, Q, and third one Q, Z, L. So, I am taking protein sequence example, taking a simplest motif here, a conserved motif which is present in kinases. kinase enzyme sites. Just like that I have taken kinase enzyme. These three are present at the active site here, and I want to see whether I can do alignment using pairwise sequence alignment or not. So the best approach will be here. So in all three sequences we have Q, and in two sequences you have D and D. Okay, D and D are there, and in two sequences I have L and L here. So my final alignment would be something like this: D gap 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 Q gap L and F D N M Q and gaps and 3 beginning gaps and QZL. So this will be my best optimal alignment. It can be achieved using dynamic programming. So how it is done here? So I have written down one sequence through one side of the cube, another sequence on another side, and another sequence on another side, and using simple pairwise sequence alignment I fill the matrices here. So I give scoring in this side 2 4 6 8 minus 2 minus 6 minus 8 minus 2. So I can use a simple same neighbor joining Needleman-Wunsch algorithm approach only. I can fill the sides here. So like this all the cells are filled here, so each cube, so suppose, but here one thing should be there, a cube have 3 sides here. So for this one 3 scores will be 3 scores for a cell in a cube because of 3 sides and 3 different types of comparisons. So there is a formula for number of pairs possible here, n into n minus 1 by 2. Since I have three sequences here, that will be 3. So 3 scores are possible here, and all these 3 scores are added up to fill the score of this small cube. So in this bigger cube here or cuboid, so each cell score will be from the 3 different sides here because of 3 different comparisons, and for 3 different comparisons we sum up them to get a one single score. So that approach is called as sum of pairs approach or sum of pairs method here. So assume that I have 5 sequences. So sequence 1, sequence 2, sequence 3, sequence 4, sequence 5, 5 sequences, and position 1, position 2, position 3 in these 5 different sequences. So what I do is I construct a pentagon made up of 5 points each indicating amino acid residue in 5 sequences here. Then I'll calculate the score of NN score, NN score, NN score and NN score. Then all these scores will be summed up and I'll give a score called as sum of pair score. Similarly for column B or second position, or column I'll take a third position here, column 3 here. So I can have NN score, NC score, NC score and CC score here. So different scores can be calculated, and the scores will be summed up to get the sum of pair score. The sum of pair score values will help us to trace the path of final alignment here. So that final alignment score will be used for deciding the similarity amongst the sequences here. One important thing about the dynamic programming is, although dynamic programming is correct approach, may be correct, but it is computationally intensive, very computationally. It takes lot of time. You require super computational sources, and it is not possible for real life sequences, hence these methods is actually not used here. There is something called as computational complexity. The idea is very simple here. So for dynamic programming here, assuming that O or computational processing time varies for each microprocessor here. So for dynamic programming, you say the time taken will be to achieve alignment of n sequences with L length given by this formula O into 2 power n into L power n here. Assume that its micro processing time is in microseconds 10 power minus 6 seconds here. So for 10 sequences each of 100 length, so it will be 10 power minus 6 into 2 power 10 into 100 power 10 here. So then we'll get 2 power 10 into 2 power 10 into, so we'll say 100 power minus 8, something like this. So it will be huge amount of time here. So if you convert this one, is it will take 2 billion years. So just to perform dynamic programming for 10 sequence with each of 100 length here, the optimal time will be billion years here. So that is not feasible in real life approaches. That's why we have to use other methods. Those other methods are called as progressive approaches here. Progressive approaches, basically they have less computational time. So we'll see about how progressive alignments will work here. So the idea is very simple. In progressive